Good day to everyone. I would like to thank the organizers for the opportunity to make our presentation. On behalf of AmbioPharm, my presentation is titled Disulfide-Rich Peptide Production at AmbioPharm, and this work is done in collaboration with our VP of Production over in Shanghai, Guoqing Zhang. The trend in peptide complexity has been increasing since the early 1980s. As you can see in the graph above, we have around 70 approved peptide drugs now commercially produced. And in the bottom part of this plot, taken from the Lau and Dunn publication, you can see that the peptide trend from the 80s to the present is showing that the number of short peptide chains being produced and developed has been going up through the 80s, the 90s, and into the early 2000s. But by the time we get to the present decade, you can see that the number of shorter peptides being developed is in the same range as the more complicated, longer peptides. And you even see some peptides greater than 30 and even a couple peptides greater than 50 residues in length that are actually being commercially produced. With that trend, additionally, the ability to produce peptides that have stable structures is also very important. And as we all know, nature has given us the disulfide bond as a way of stabilizing structural elements in peptides and in proteins. Peptide drugs that have multiple disulfide bonds are shown in this particular table. Zacconatide is the first multiple disulfide peptide drug developed. It's a peptide that's 25 residues long with 3 disulfide bonds. It's originally isolated from Conus magus, which is a cone snail. And this particular peptide is an omega chronotoxin, meaning that it binds to calcium channels, and it's actually used to treat intractable pain. And because of its way of being administered, which is an intrathecal implant, that makes it much less desirable and is only used in very rare special cases. And thus the continual need for this is about 50 to 100 grams per year. Linaclotide is another tri-disulfide containing peptide. This peptide is only 14 residues long with 3 disulfide bonds. It's a heat-stable neurotoxin analog. It's used to treat chronic constipation and opioid-induced constipation. And it interestingly is delivered by an oral route, making it a very convenient way of managing these type of situations. The current annual need for this is about 80 kilos per year that is projected. In a similar manner, plecanatide is a uroguanilin analog, which is very similar to heat-stable neurotoxin. It's used to treat the same type of conditions, chronic constipation. And basically, it's a 16 residue peptide, but it only has 2 disulfide bonds. And the current annual consumption of that is approximately 100 kilos per year. We'll talk a bit more about this one later on. There is a unique mechanism of action for this that helps relieve some of the symptoms associated with taking these types of peptide laxatives, if you will. A protein probably represents one of the most complicated peptides synthetically that has been produced. It's also known as bovine pancreatic trypsin inhibitor. And this particular peptide became important when the prion issue was hitting back in the early 2000s in mad cow disease. There was a desire, instead of isolating this from bovine pancreas, to make it synthetically, and it has now been done by Bachem, and it's produced currently, we estimate, around 5 kilos per year. This peptide is 58 residues long and 3 disulfide bonds, and it's used to control bleeding during surgery. Of course, very famous molecules are the insulin analogs such as Humulin, Lantus, Toujeo. These peptides are also 3 disulfide bonds, but they are 2-chain peptides that have 2 intermolecular disulfide bonds and 1 intramolecular disulfide bond in the A chain. Due to the need for these for people that are diabetic, these are produced in very large quantities, currently all recombinantly, in metric ton quantities on an annual basis. Now, there are a number of peptides that are still in clinical development or close to reaching clinical development. Some of those include Disulfide, which is being developed for autoimmune disease. This is a 3-disulfide peptide originally isolated from a sea anemone, and then analogs were developed to make it more specific. It was being developed by Aramid and Canada. Now it's being developed by a new company called TEKV, and this particular peptide should be entering phase 2 clinical trials later this year. Hepcidin, LJPC401, is basically human hepcidin used for treating beta thalassemia. This is a 25 residue peptide with 4 disulfide bonds. This particular peptide has now stopped in clinical development from La Jolla due to some issues in the clinical trial where there was no real effect over placebo for beta thalassemia patients. Another interesting peptide also with 4 disulfide bonds is Chlorotoxin, or Blaze 100, which is basically a chlorotoxin dye called Disulfide. And this particular peptide is in phase 2-3. It's basically used for imaging cancer metastases to allow surgeons to excise all the tumor that they may not see well with their naked eye. Isoganin is another peptide that was being developed as an antimicrobial, also called protegrin. This peptide had 2 disulfide bonds. It was being developed by Intrabiotics. They ceased development. Hebrew University then took it up, and I think it's still stuck at the same stage around phase 3. More recently, Canada has been developing a KCP400, which is an alpha-conotoxin analog. This particular peptide is also used for chronic pain treatment using a novel mechanism of action, such it hits the nicotinic acetylcholine receptor. It's got 2 disulfide bonds, and that's now scheduled to move into phase 1 clinical trials probably in November of this year. T20K is a cyclotide, one of the type of molecules David Craik has made famous. This particular peptide is being developed as a potential treatment for autoimmune diseases such as multiple sclerosis. It's got 3 disulfide bonds, and it has the head to tail cyclic structure, so it actually has 4 levels of constraint, being developed by Cyclone. Monash is developing another scorpion toxin called HSTX1 with an arginine substitution. Autoimmune indication, preclinical. It's a 4-disulfide peptide, and it's supposed to be entering into preclinical development sometime in 2021. The COVID crisis has kind of delayed that one somewhat. Lastly is another very interesting molecule originally isolated from a tarantula. It's called GPTX1. It's interesting that it binds to a novel sodium channel called NAV1.7 and may be useful for also as a pain therapeutic. It's currently being developed by Amgen, much like Protoxin and the related kind of compounds. It's got 3 disulfide bonds. Now, where are the problems in making these kind of peptides? The solid phase strategy improvements for making long peptides that have multiple disulfide bonds include use of special derivatives called pseudoprolines or even dipeptide building blocks to help break up resin aggregation effects. We can also use specialty resins such as PEG resins, Tentagel or clear or related resins with lower substitution to help minimize steric hindrance of the developing and buildup of the peptide in the solid phase. Next step is whether to use an orthogonal protecting group strategy, which protecting groups to choose in that strategy, or do you choose a random oxidation, or as I like to call it, God-assisted folding. Is he helping me today, or is he hindering me today? Hopefully the most favorable isomer folds is the correct one, and that's the logic that's used for a lot of these 3 and 4 and even 5 disulfide peptides that are being developed. Next, do you pre-purify that linear peptide, or do you take and fold the crude linear peptide directly? When you have multiple disulfide bonds, you have the potential to develop isomers according to the formula that you see above. P is equal to N prime divided by, or N factorial divided by N over 2 factorial times 2 to the N over 2. What that basically means, by the origami snake below, do you get 3 isomers with 2 disulfide bonds? 3 disulfides gives you 15, 4, 105, 5, 945. So accidentally, are we going to get the right effect and get the right peptide isomer? We hope so. We hope it's a serendipitous experiment like making chocolate milk. If we choose an orthogonal route, our selection of protecting groups basically for one disulfide is using cis-tridal. Tridal comes off in your standard TFA-D protection, and you can make the disulfide bond by either titrating with iodine or using a peroxide oxidation type format at around pH 7.5 to 8. 2 disulfide bonds, one disulfide can be positioned with cis-tridal pair, and the second disulfide bond with acid-amidomethyl or ACM. ACM can be taken off with iodine, and tridal can be oxidized, as we just mentioned, with peroxide. 3 disulfides, we would choose cis-tridal, cis-ACM, and cis-MOB, methoxybenzyl. Methoxybenzyl can be taken off with a stronger D protection, such as TFMSA. There are some reports out there about taking it off with TFA in the presence of DMS. That has not been successful, at least in my hands. With 4 disulfide bonds, you have the ability to have cis-tridal on 2, cis-ACM on 2, cis-MOB on 2, and a new protecting group called 4,4-dimethylsulfonyl benzhydryl. This particular protecting group is also called the safety catch protecting group, and that gives us a 4th level of orthogonality, and we successfully made human hepcidin, helping to prove the disulfide pairings that were reported initially incorrectly and subsequently corrected by Amgen. And this helped establish that indeed those were the correct assigned disulfides. More recently, there's another disulfide pair possible with the nitroveratril group. This particular protecting group, ONV, is photolabile, so maybe it would be possible to even do 5 disulfide bonds orthogonally. But what you run into when you do these and you do the intermediate purification steps in between is you kill your yield. So from an industrial scale-up perspective, going beyond 2 disulfide bonds, we really have to rely on Mother Nature to help us fold to the correct isomer. In our case studies, the first case study that we want to talk about that we've produced at AmbioPharm is the generic peptide called Eptifibatide. This particular peptide was originally developed from a toxin from rattlesnake. It basically is a short peptide with a single disulfide bond that has an RGD motif that Core Therapeutics then substituted that with homoarginine instead of arginine, and they were able to develop this particular drug, also eliminating one of the cysteines to reduce proteolytic stability. To increase proteolytic stability, they used MPR instead of cysteine. This particular peptide, we produce it with the tridal protecting groups on the cysteine and the MPA. The crude linear product is pre-purified and then subsequently folded using an iodine titration. The chromatograms that you see here represent the crude linear, followed by PrepH PLC. You can see a very nice looking product, greater than 96%. Following cyclization, single peak transformation, easily purified to greater than 99%, mass spec showing the correct product in the positive mode. Plecanatide is a peptide that we produce in very large quantity at AmbioPharm. This particular peptide has a unique feature to it. As we were talking about heat-stable enterotoxin, linaclotide, and uroguanilin. Uroguanilin has a phenomenon where it can spontaneously inactivate itself according to pH and temperature. This actually is beneficial in the development of the drug because once you take it, it basically will spontaneously inactivate as it traverses through the gut, and it lowers the amount of product that actually is still active by the time it gets to where it needs to cause its effect, which is the large intestine. When it gets there, it lowers the ability to cause diarrhea, and therefore it's an alternative to Linzess, where the major drawback to Linzess is very bad diarrhea. We basically have a strategy to produce this peptide by 3 fragments. It's a hybrid strategy to assemble the 3 fragments into the 16 amino acid sequence. We use the cis-trityl at positions 4 and 12 and cis-acetamidomethyl at 7 and 15. This just shows you the linear crude product oxidatively folded with peroxide at alkaline pH. We get the monocyclic product with still 2 ACM protecting groups on the second pair of cysteines. Following treatment with iodine for 2 hours and ascorbic acid reduction to treat the iodine and turn it into iodide, we end up with the dicyclic product that you see above here. The active A isomer is the major product. We do see some of the B topoisomer form just because of the length of time that it takes to do the oxidation. This is easily removed through purification, reverse phase, and you can see that we have a product here with a purity greater than 98% TFA method, 97% using TEEP, and the mass spot on at 1681 in negative mode. Another example would be the Linaclotide. As we mentioned in the previous molecule, it's also a GCC agonist used to treat irritable bowel syndrome, chronic constipation with an oral route. This particular peptide has 3 disulfide bonds. We chose cis-trityl for all 6 positions. We once again pre-purify the linear, and we go with our God-assisted folding using glutathione-based redox pair. As you can see here is the overview of the process: reverse phase, purify the linear, lyophilize, refold the linear, and then take that and purify. The chromatograms correspond to each one of those steps here. You can see that the crude product following folding has a relatively good purity level of 85%. And this is easily purified by reverse phase to greater than 98%. And we once again have the right mass as indicated by the negative ion mass. Next is chlorotoxin. This particular peptide is quite complicated. It's on the longer side, 4 disulfide bonds. It's a 36 residue peptide, C-terminal amide. It also has 2 methionines located close to the N-terminus and another methionine right there around position number 13. So with all these methionines and cysteines, following the synthesis, which basically uses a rink amide linker, cis-trityl at all the positions, pseudoprolines were substituted in and dipeptide building blocks at strategic positions to improve the crude quality. Following TFA cleavage, it's necessary to do the ammonium iodide DMS reduction step to help eliminate methionine sulfoxide that forms during the synthesis. We take this product straight crude, dissolve it in oxidatively folded alkaline pH, and use a redox pair of glutathione to help facilitate the folding. You can see the linear crude product, reasonable purity. Following cyclization, we basically have a purity of around 20 to 30% in that range. It's purified through several different steps to get the final purified chlorotoxin. Purity was greater than 96%, and as you can see, the corresponding multi-charged ions demonstrate the correct folding with 4 disulfide bonds being formed. So in summary, I just want to say that, we routinely do cGMP synthesis at AmbioPharm of complex disulfide-rich peptides. We have produced commercial synthesis of plecanatide at quantities exceeding 100 kilos per year, and we use relatively innovative approaches to handle large-scale production, including solid phase, hybrid, and classical synthesis at AmbioPharm. And for those of you that aren't familiar with AmbioPharm, AmbioPharm is a unique business model where we are headquartered in the USA with our manufacturing synthesis labs over in Shanghai. The North Augusta site in South Carolina, we can do purification, isolation, lyophilization, QC release, special cyclizations, things of that nature. But all of our chemistry is done over in Shanghai, which is where the site for all the raw materials are. And it makes it very easy in terms of dealing with the synthesis by lowering the cost of key chemicals, solvents, and helping pass those along to savings to our customers. We are having an expansion over in Shanghai that should be finished by the end of the Q3 2020. It includes a major administration building that you see on the lower portion of the slide here with 3 workshops. These workshops will be handling solid phase, solution phase, and purification aspects, as well as quality control. We have one building dedicated to hydrogenation that you see. That's a small building towards the back of the campus, as well as solvent recovery. And this will allow us to have a much better green footprint and help improve our economic impact for producing peptides. The Shanghai completion, as you can see, is going on. I was there in 2019 and then would have been there in 2020 in March, but COVID kind of prevented that from happening. So I got photos just to show how the project is progressing over in China. Similarly, we have completed an expansion in North Augusta, and this is a 56,000 square foot manufacturing building where we have new 45 centimeter column, 60 centimeter column, 100 centimeter column that's being installed as we speak, and a new 1,000 liter lyophilizer to expand our capacity. What we have in terms of peptide manufacturing capability, we believe that we have the largest overall manufacturing capacity in the world, with multiple large-scale reactors up to 3,000 liters in solid phase and 5,000 liters solution phase. We have purification equipment spanning from 5 centimeter all the way up to 100 centimeter. We have multiple lyophilizers for all the way from 100 liters all the way up to 1,000 liters, as well as a few manifold old-style lyophilizers. QC lab that's set up to handle all the standard release parameters and specifications that we must meet in any of our GMP production. We have currently increased over the past several years from 2015 to 2019 from 66 kilos output to 2019, we hit 213 kilos of finished product that was released. AmbioPharm is a commercial manufacturer of cGMP grade peptides. We're eager to work with any partner that needs our type of service. We routinely handle very complex disulfide-rich peptides from inception milligram quantities all the way to commercial launch greater than 100 kilos. We are uniquely positioned to offer exceptional service for very economical rates due to our business model. Please, if you have any questions, I'll be happy to take them. And I thank you for your attention.